Our next speaker will be Rhishikesh Bargaje, and he is going to give us a talk with the title of Single Cell Analysis of Cell Fate Branching, from Computational Pattern Recognition to Cell State Dynamics. Thank you.
Thank you. Okay, cool. So good morning. Thanks to the organizer putting together this event, annual event, and bringing us all together. And also thanks to Sui, because he was supposed to give a talk. And thanks to the unscheduled travel. It's not usual that postdocs get a chance to present at such a nice conference. So what I'm going to do in the next 20 minutes or so is try and talk to you about a novel approach where we have tried to analyze cell state transitions to study differentiation at a single cell resolution. Most of you know the holy grail of regenerative medicine is that I can draw blood from any one of you, add a few proteins to make pluripotent stem cells called as iPS cells, and be able to differentiate them to any cell type in your body type, in the body. So then I can use them for screening, as well as I can transplant them in people so that I can get a functional heart, as Chuck gave a nice example just a couple of talks back. And we've used gene expression as a guiding principle. So we have used microarrays or next-gen sequencing to guide our differentiation protocols. And of course, understanding the gene-regulated network behind what drives the gene expression profiles is very much important. Also, what we know is that a stem cell, a pluripotent stem cell, has to go through the series of binary branching events to give rise to the diversity of the cell types. And Waddington very famously in 1950s put forth this epigenetic landscape as a metaphor to understand this process. How does a stem cell differentiate into mature cell types? And lately, with all this advent in the technologies where we have used single-cell tools, and we have created these single-cell analysis methods to sequence genes, proteins, microRNAs, and even to look at metabolite at single-cell resolution. So people have tried to look at, study the process of differentiation or even de-differentiation in some cases. And eventually, again, as I said, tying it back to the gene-regulated network that governs this process. So we came in with a very simple question in mind. That is, if a stem cell differentiates into, let's say, a cardiomyocyte or a hepatocyte or a neuronal cell, what are the different cell states it goes through? So for this, to study this kind of phenomena, we essentially used very simple systems. One is the cardiomyocyte differentiation system. Actually, the protocols were developed in Chuck Murray's lab, where you give certain embryonic cues to the pluripotent stem cell, and by day 15 or so, you get beating cardiomyocytes in your dish. So some of the first days of differentiation are, of course, very much important and play a major role in the efficiency of differentiation. So we profiled single cells for the first 5 days using various phenotypic markers. So as I said, like cardiomyocyte differentiation is a very well worked out system. So we know which cell surface markers are expressed on cells at different stages of differentiation. So you can label these cells with the fluorescent antibodies and sort them into a 96-well plate for single-cell qPCR. So at the end of this entire effort, we have about 3,000 individual cells for which we have gene expression profiles for 96 genes, as well as cell surface or with the phenotypic information for all of them. So now the first steps that we do is use a non-linear dimensionality reduction step where, so this is a 96-dimensional data because you have gene expression profiles for 96 genes. So you compress it into these 3 dimensions. And what you can see is that the stem cells form a tight cluster. But once they start seeing activin A, they start to migrate towards the next stage of differentiation, which is much more like primitive streak-like stage. And once they start seeing BMP4 and chiron, they split or they bifurcate into either an endodermal lineage or a cardiac mesodermal lineage. And of course, we know all this because we know which genes are expressed at what stage of differentiation. So to go back a little bit to the complex systems theory, so phenomenologically, if a cellular system is undergoing a critical transition, here is what you should see. So as I said, a stem cell, which is a very homogeneous, stable cell state, it's in a deep well of attractor. But when you come up with an external stimuli or a signal, as I said, in our case, activin, you should destabilize this attractor, meaning the well of attractor should become shallower. That means there will be increased cell-to-cell variation. And if you come up with a sustained external signal, it should eventually bifurcate into two different cell states. And if you see this, you should see a couple of points. One is decreased cell-to-cell correlation at around the tipping point, because you've the well of attractor is shallow, so the cells spread out on this attractor. And secondly, you should see increased gene-to-gene correlations, because the cells that are being driven into the next lineage start expressing certain genes that coordinate that particular movement. These are anti-correlated to the genes that will coordinate the movement to the other lineage. So you should see increased gene-to-gene correlations. So just to summarize what I said with critical transitions, so we should come up with two testable predictions with this understanding. One is, OK, I'm going to go a step back. So traditionally, we used to do a lot of microarray studies where, let's say, you had treated versus control. So you crush the cells, prepare RNA, put it on a microarray. All you used to get is one single vector. You get one column of data. But now with single-cell analysis, you get a matrix, which is a statistical ensemble of a high-dimensional system. That means you get a matrix of data for a sample. And that it lets you calculate what Sui's lab has pioneered. It's called as index of critical transition, which is gene-to-gene correlations divided by cell-to-cell correlations. So you should see an increase in critical transition when the cells get destabilized right before the critical transition. As well as you should see this rebellious cells or undesired cell types in your dish. So what happens in case of erythropoietic differentiation? So this was done in Sui's lab, where you take progenitor cells, the EML cells, and give them EPO. And you should expect erythroid cells in your dish. And the cells are labeled with SCA1, which is a stem cell-specific marker. So when you do a bulk analysis, you see decrease in expression of SCA1. But what happens at the single cell level? So you do a FACS for these cells. So at day 0, you have a very high expression of unimodal expression of SCA1. As they start seeing more and more of EPO1, you get one branch, which is SCA negative, and that's the one that is going to become erythroid. And you get a second smaller distribution of SCA positive cells, and these are what we call as rebellious cells. And these are not, although they have received the signal, they don't differentiate into erythroid lineage. And of course, it really gels well with the theory and the physics behind pitchfork bifurcations. And again, in ecology and even in physics, these systems are very well worked out. And eventually, we can also tie this to the gene regulatory network behind this kind of behavior. In this case, again, since it's a very well worked out system, we know which transcription factors, the GATA1, PU1, that is driving this process. In cardiomyocyte differentiation, we see the same thing. So here, unfortunately, this is like opposite way. So this is day 0 here and going back to day 6. So you see at around day 2.5 to 3, there is a bifurcation in the system for CKIT expression. And these are the cells that are going to give rise to the endodermal lineage. These are the rebellious cells. So again, like going back to the erythroid and myeloid differentiation. So what they did in Sui's lab is to use these progenitor cells, give them EPO to get erythroid, or GM-CSF to get myeloid differentiation. And again, as I said, this system is very well worked out, and we have these two testable hypotheses. So they captured about 600 cells and looked at about 20 genes. And so here's what it appears in a 3-dimensional PCA plot. So these are the day 0 untreated cells. Once they see some kind of treatment, you have a different they move into a different cell state. And as you continue to give the cells with the external stimuli, they'll separate more and more into either the erythroid or the myeloid cell state space. So in order to look for critical transitions, as I said, like you should see decrease cell-to-cell correlation. So at day 0, when the cells are all similar to each other, you see really high cell-to-cell correlations. But as they approach a critical transition, you kind of see that there are two subpopulations that emerge. So there is a very high correlation between these cells and high correlations between these cells. At the gene-to-gene correlations, you start seeing that there is right now just one big blob, whereas at day 3, you start seeing there is a set of genes that are correlated and set of genes that are anti-correlated. And in terms of the index itself, you see a very low index early on, and right before the critical transition, the index goes up. So you know when is the critical transition taking place with this kind of measure. And for the cardiomyocyte differentiation, so you see somewhat similar results. So that is, for gene-to-gene correlations, the average correlations hover around 0 because there is no active regulation of genes except the ones that maintain the stem cell state. All of the genes are only differ by noise. So you have majority of the correlations hover around 0, and you have very high cell-to-cell correlations. As you approach the first transition, you see decrease in cell-to-cell correlations, as well as increase in gene-to-gene correlations. And you see the similar picture for the second transition. Again, in terms of critical transition index, you have very low index for extremely homogeneous stem cell population. The index starts to increase. There is a critical transition to primitive streak-like stage. This index drops. Another increase, and drops when it commits to the next step of differentiation. And we have now validated this with several other systems, including publicly available data sets. So this is a lung differentiation, in vivo lung differentiation, where they did collect single cells, looked at the single cell RNA-seq. So we took out 2,000 genes, and we can still see the same behavior. So when cells approach here, you see increase in the IC value. When cells make this commitment, you see another increase in IC value. So the importance of this method is that it's mechanism-free. You don't need to know which genes are involved in this process. And that's the beauty of this process. But still, you can drill down and try to identify the genes that are correlating or anti-correlating to identify some sort of mechanism behind this process. So here is an example of that. So what you see here is gene-to-gene correlations for transcription factors that are important at several stages of differentiation. So NANOG, a well-known transcription factor for stem cells. T, EMS, GSC are involved in primitive streak-like stage. MESP1, HAND1 are cardiac mesoderm, and SOX17 is endodermal lineage. At the beginning, when they're all stem cell-like, there is no active regulation of these genes. So all you see is like a big, giant blob with no correlations at all. Whereas when you approach the first transition, which is at around day 1.5, where they go from epiblast to primitive streak-like cells, you see active correlation of genes that are involved in this process. Similarly, when you approach the second transition, that is, you go from primitive streak to either cardiac mesoderm or endodermal lineage, you see that the cardiac mesodermal are regulated together and show really good positive correlation. And they're anti-correlated to the SOX17 gene expression, which is the endodermal lineage. So you know what is the governing or the mechanism behind this decision-making process. So next, what we did was to use cell surface marker as the axis of differentiation. So in our case, we were lucky that, and again, CKIT is a very well-known molecule in cardiac differentiation field. So it's a stem cell marker. So you get a unimodal distribution of CKIT at around day 0, 1, when cells are stem cell-like. And when you approach this 2.5 to 3 stage, you get a bimodal distribution where the low CKIT cells are going to be cardiac cells, cardiac mesodermal cells, and this is going to be the endodermal line. So what we did here is to collect single cells from different parts of the distribution. So you have CKIT low cells, medium cells, high cells, sort them out into individual 96 well plate, and perform single cell qPCR for the same 96 genes. So what you see here is for day 0, where all the cells, like either whether it is low cell, medium cell, or high cell, there is a very high correlation between all cell types, meaning there is no knowledge or there is no information in this distribution. But as you approach the critical transition where the cells are going between either mesodermal or endodermal, you see that low cells are alike, high cells are alike, but there is not much correlation between low and high. That means there is now information in this unimodal distribution. So cells that are here are different than cells that are on the other end of the distribution. So the cells here are going to become cardiac mesodermal. Cells at this end of the distribution are going to become endodermal. And of course, we can confirm this with certain genes. So NANOG is a well-known stem cell marker. So you have very high expression. As it differentiates, it goes down. SOX17 and HAND1, which are imported in SOX17 for endodermal, HAND1 for cardiac mesoderm. You have no expression of them. You start seeing some noisy expression of these two transcription factors. But once the decision is made, it's almost black and white. So now we have gone a step further and tried to use this kind of information or knowledge to make the differentiation process better or efficient. So what we did is to create a matrix where we have different concentrations of Wnt inhibitors and different concentrations of BMP4. So you have 15 different conditions. So we differentiate cells using all these 15 different conditions, go up to day 20, and look at the total number of cardiomyocytes that you get in the dish. As well as, since we know that one of the major critical transition happens at around day 2.5, so you also collected about a small population, hundreds of cells at different CKIT distributions at day 2.5 and performed qPCR for all, again, these 96 genes. And what we can what we can construct here is a classifier, a random forest-based classifier, to predict the efficiency of differentiation. So essentially, you can capture cells at day 2.5, measure expression profiles for all these 96 genes, or in this case, probably just 3 or 4 handful of genes, and predict what will be the efficiency at the end of day 15 or day 20. So if you're coming from a commercial background, it's a huge save on time, effort, and money. And of course, this is in our system. It probably needs to be validated in many more systems. And as, again, Chuck can probably validate, if you go from line to line, there is always, you have to always basically come up with try out all different conditions and standardize your method, the treatment, so that you get efficient differentiation. So this approach can make it even much more faster. OK, so just to reemphasize that since we know this is where the cells make the decision, we can probe the system very early on to get efficient differentiation later on. So here's another decent application of such system. So as I said, SOX17 is involved in endodermal differentiation. So one can come up with an hypothesis where I inhibit SOX17. So all I should get in my dish is cardiac mesodermal cells. So fortunately for us, somebody in 2007 did this experiment where they inhibited expression of SOX17 by using short hairpin RNAs. Unfortunately, they didn't get any cardiac mesoderm in the dish at all. And what they end up saying is there are cell non-autonomous factors that we don't know are important for this kind of process. Fortunately for us, what we find is that we had a receptor-ligand pair in our 96 genes set. And what we can find is this is the mesodermal branch, and this is the endodermal branch. PDGRF alpha is a very well-known marker for mesodermal branch. So you see higher expression of this receptor in the mesodermal branch. But guess where the ligand comes from? It comes from the endodermal branch. So you can come up with an hypothesis where you can inhibit SOX17, but still provide PDGF alpha, the ligand, from outside to make efficient cardiac differentiation process. So just to summarize here, and I hope I'm in time, within the time limit, is that we've used theory of a complex system to come up with a critical transition index that can help you come up with two different testable hypotheses. One is the increase in critical transition index and presence of rebellious or undesired cell types in your dish. And we would eventually like to tie all this back to the gene-regulated networks and hopefully make all the different cell types in the body, as well as make this process of differentiation efficient and faster. Okay. And so Callie is the one who has done a lot of computational analysis for the cardiomyocyte project. Mitra is the one who did a lot of the erythropoiesis work, along with Joseph. And so I would like to acknowledge, of course, Lee Hood for and Sui Huang for the project. Yeah, and some of the folks, of course, from Ice Cream and funding agencies. Thank you.
Thank you, Rishi. Let's take some questions.
Thanks. Thanks. That was really cool stuff. I'm trying to wrap my head around the assumptions you need for this model about cells approaching a bifurcation event. It seems clear a priori that if you are getting close to it, that the variance across cells must increase. Mm-hmm. But it seems like in order to get that the gene correlations must decrease, that there must be some other assumptions required. I mean, you can imagine that there's only a handful of genes that are involved in this bifurcation, in which case the overall correlation profile might not change that much, or there could be some other process that causes gene-to-gene correlation to deteriorate. So can you say a little bit more about what's necessary to make that that prior true?
So, I mean, we do believe that there is one universal gene-regulated network for entire, like all different types of genomes, right? So even if there are certain number of genes that are needed for making this like jump into either lineage A or lineage B, since they're related to the link to the other genes, you still get enough change in gene-gene correlation. So I didn't even show the hepatocyte differentiation. We observe the same thing. We have done it for neuronal differentiation. We see the same thing.
Okay, thanks.
Is it okay? All right. So I want to follow up on the question of whether you can infer causality when you assemble sort of these hierarchies. And KIT came out to be a very predictive molecule for for distinguishing endoderm versus cardiac fate, for example. Does this in any way imply a role for this molecule in the signaling pathway that controls differentiation, or is it simply algorithmically useful?
It's right now at this stage algorithmically useful, or it's a good readout. But in terms of mechanism, probably you need to look at gene-gene correlations at least at the transcription factor level to really get behind the mechanism of it.
And just a quick follow-up, are cells really rebellious, or are we just sometimes confused about biology?
Yeah, I think I'm humanizing them by calling them rebellious.
Very cool talk. Have you tried to build pseudo-time trajectories for your cardiomyocyte differentiation and so on? And do you see the same branching events that you were seeing using your 3D PCAs?
Yes. So we have tried, like one of the popular methods is called as monocle, that is from Cole Trapnell's lab. And I might get a flag for it, but so at least some of the first versions were based on a linear method, and we believe that the cellular systems are non-linear. So that's why we kind of didn't use it, but we have done it using like monocle, MDS, PCA. You still get the same structure. It's just that if you use monocle, it tries to make a continuous trajectory, which is actually not true, because you get these stable states. It's not. Essentially, by using such kind of methods, you're enforcing a structure. You're enforcing like all similar cells to be together, and it makes it appear like a continuous process, but it is not a continuous process. But we have tried it, and you still get similar results. Okay, thank you so much.
Okay, thank you.